Good morning, everybody. Welcome to this workshop. Nice seeing all of you. We're still expecting some other people to join us. And I can see many people also joining online. So we just start. Sorry for starting now. We should have started at 9. You know, some people use this African time. I don't know whether you know about that. You know, something is supposed to start at 9, they add 30 minutes. I will join them later. So once again, welcome to this workshop. I'm excited to see all of you. Today, we have this workshop on qualitative and quantitative research synthesis, and we're just talking about systematic review, meta-analysis, and science mapping. I'm so excited about this workshop because I've been able to see the potential of this workshop. So what I just want to quickly take you through is to talk to you about the importance of this workshop, and we're also going to be talking about what we are going to be doing today, the people that will be lecturing us, our facilitators. Then I also want to talk about what I want to be the outcome of this workshop. And so let me briefly talk about the importance of this workshop. Qualitative and quantitative research synthesis, including systematic review, meta-analysis, science mapping, are increasingly being applied to advance our understanding of different phenomena, research trends, increase statistical powers of scientific evidences, and identify research gaps for future research directions. These approaches are easily implemented in commonly available open source and commercial software, even with no computational skills. Participants at today's workshop will be taught the various skills and step-by-step procedures involved in applying these techniques to their research and increase their capability to publish in high impact journals in any discipline. Why am I really excited about these techniques? I believe that with these techniques, you can even do research when the lab is not available. You remember during the pandemic, we couldn't work in the lab. We wanted to work in the lab, but it was not possible. We're told to be at home. And let me tell you something, another pandemic is coming. I was listening to Biden that was saying that we've got to prepare for another pandemic. So we are not going to go back to pre-2020. So we must keep on understanding that these viruses are evolving. And I like this type of techniques. Even when you are not in the lab, you can still work. And sometimes, even not during pandemic, like at DUT, we have this problem with requisition is terrible. We need to really work on that. It takes forever for your for your research materials to come. But with these techniques, you can begin to do certain things and still be able to have research output. And I also like what my HOD said. My HOD said, Professor Ferroswala, meta-analysis can be considered as part of fourth industrial revolution as it signals a change in paradigm on how we view research data, profound. So it's very, very important as we begin to talk about the fourth industrial revolution, we must begin to think about what are the skills that are very, very important. We talk about blockchain, we talk about artificial intelligence, all those things, but this is also thing you can also learn. Meta-analysis, systematical review will always help you to have a kind of hedge in your research. Let me quickly talk about the people that will be speaking to us today. There are people that they know what they are doing. They have mastered the skill of science mapping, systematic review, and also meta-analysis. And you can see from their research output, Dr. Ekundayo, and also Dr. Olasende, you will see that they've published some articles there about how they have been able to use these techniques we're talking about. In 2022, we have published articles, and also Dr. Ekunda will be presenting at IUFOS, International Union of Food Science and Technology in Singapore later this year. We'll be talking also, how he was able to apply the technique we're talking about. And these are all the manuscripts under review that as a result of these techniques that we're really talking about. Let me quickly show you the program of event for today without wasting time. I'm doing the introduction now, and Dr. Olasende will be doing the introduction to the common quantitative and qualitative research synthesis, approaches, and software. 10:30, we're going to have a tea break. Then from 11, we're going to have effective ways to acquire data sources from different scientific databases. 1 to 4, we're going to have our lunch, and by 2 to 2:30, we're going to be talking about data extractions from articles and scientific graphs, performance of statistical analysis, and guide for interpretation of results. Then later on, we're going to have discussions talking about questions and answers, and then we close. I really want us to think about something. I've been thinking about what do we really want to achieve from this workshop. One of the things I think I really want us to achieve is, at the end of the day, I want us to have a kind of research group. We don't want it to end there, because when it comes to meta-analysis and systematic review, you need skills from different professionals, different people working in different area of research interest. And at the end of the day, I want us to be able to come up with a group that we can work together. And Dr. Ekunda is going to tell you how we're going to, come up with those groups. People that will be able to respond. Maybe we're going to give you an assignment to do. People that are able to do the assignment, we can come up with a research group that we can work together, publish together. And we don't have to be in the same lab to publish together. We can be all over the world, and all of us can come together and publish together. And that's how we can solve problems. We have messy problems in the world, and it requires system thinking for us to solve it, to solve these messy problems we have in our world. So, and that's what I'm really thinking about, that I'll be able to do. And I also want to talk to you. You might not be able to grab everything these guys are going to talk about today. They've been doing it for months. So don't really crucify yourself if you don't know everything they are saying. But at least you've been introduced to it. It's going to be recorded. You can go back to the recordings, check, see you are able to understand. And so don't really crucify yourself if you don't get everything they talk about today. Last but not the least, I want to acknowledge NRF. Without their support, we would not have been able to organize this type of thing. And I'm grateful for their support. And that's it for me. And I'm going to be inviting Dr. Ekunda. I think okay, Dr. Olasende, be the one speaking. I need to make you the host. Yes, okay. Thank you. Okay. Okay, that's fine.
Recording in progress.
Okay, if you're joining us online, just try to mute your mics so that other people can hear clearly. Just share my screen. Sure. All right. I know this is not tenable, but let me just start by apologizing for coming a bit late. It was an unavoidable, unscheduled appointment. So I had to attend to a microscopy appointment, which was rescheduled and beyond my control. So I do apologize. I know it's like an African time thing. So, but here we are. Before I go into the presentation, I want to appreciate the organizers of this workshop. It's a great privilege to be here, and I'm so happy to be here. I'm glad, and I want to appreciate Prof. Prof has been a good motivator and support. I met him I've heard about him, but I met him first time, I think that was in 2018, and took us out for a lunch, and that was how we started the chat. And he's been doing great works, encouraging young people, early career like us. And he's been a supportive pillar for young people. Prof, we appreciate you. We thank you for the work you are doing, the platforms you are creating. You see his post on Facebook, teaching people, encouraging people, books and all of that. We see your works and we appreciate everything. So here we are today for the workshop on qualitative and quantitative research synthesis approaches and software commonly used. So don't be afraid by these funny words. It's just it can be a deception. So don't think you can't know it. So permit me to use this. Let me burst the bubble. I'm not an expert, but I can do research synthesis. I can write narrative reviews. I can write systematic review, and I can write meta-analytic reviews. So you can also do the same if you want to, and that's why we are here. The goal of this presentation is that, or this workshop is that at the end of this workshop, you will be able to write a narrative review or systematic review or meta-analysis at whatever level you can. And let me start. Put off your audio. Okay, let me start by asking that how many people have written a review at one time or the other? Any kind of review. I know everybody has written literature review at any time. Maybe narrative review, anyone? Okay, narrative 1, 2. Systematic review, anyone? Okay, 1, all right, that's great. Meta-analysis review, anyone? Okay, so I know the large audience of people I'm speaking to this morning. So it means, and I want to tell you that you have not written a systematic review for meta-analytic review does not mean you can't write a review. I was in a meeting one day and somebody said, "Can you fly a plane?" And I was like, "I'm not a pilot, so why would I fly a plane?" But he told me, "Do you know you can fly a plane?" I was still wondering, how can I fly a plane? But he said, "You can fly a plane." So I came to my senses and I said, yes, I can. If you have thought how to fly a plane, you can. It's possible. So you can write a review, you can write a systematic review, you can write a meta-analytic review. All right. So the point is, at the end of this workshop, after this workshop, that you will write any of these reviews will be determined by you. And it will be important that you are able to go back, do some, because we will be talking about the basics, the nitty-gritties. We won't be able to go in depth because it's diverse, is huge. And we are here in different areas, maybe humanities, biomedical research field, and different kinds of field. So we can't delve into much, but we can talk about some nitty-gritties, some basic things you need to look out for. By the time you sit down, go through some resources, some references we will point your attention to. I tell you that you can begin to develop ideas in this direction. Now, like Prof have said that you don't need to get into the lab to do this. So it means that you can publish as many papers as you want without getting into the lab by writing these reviews. So I'll be talking around this listed outlines: research synthesis, qualitative, quantitative synthesis, critical and common review approaches that we have, and then I will stem it down to systematic review and meta-analysis. I will touch on science mapping because we are also revolving around those three. We're talking about qualitative and quantitative research around systematic review, meta-analysis, and science mapping. So I'll touch on science mapping, which is very cheap. You can easily grab, and you can do one or two things. And we'll talk about common software and tools that can be used to do all those things. So, and I'll be asking questions. So I know you ask me questions, but I'll ask you questions to get your attention and to know that, to have an interactive time. But please feel free to point my attention at any time. You can signal by raising up your hand, and I'm happy to draw back. If I can't answer, we have experts here that can answer. You know, Dr. Kundayo actually sensitized me to this approach. I wrote my first review in 2017, and it was a narrative review. I didn't know anything about bibliometric analysis, systematic review, or meta-analysis. So he just sent me a message one day and said, come, we can do this. And we brought out a research question, and he didn't teach me. I was expecting that we will have a Zoom meeting and he will say, do this, press this, touch that, you know. I told him, let's have a Zoom meeting and take me through. He said, no. He gave me resources, read this, go through this, and I began to search. I'm yet to finish reading them, but I have better understanding and I'm better off now. All right? So you can do the same. We will point your attention to those references. If you can sit down to synthesize them, to read through, and to get how to do it, you can. All right? So you can tell yourself, I can. So let's talk about research synthesis. It's a process of bringing together data from a set of included studies, meeting pre-specified inclusion criteria with the aim of drawing conclusions about a body of evidence. Take note of the underlying words. They are very critical. Bringing together included studies, pre-specified inclusion criteria, and you'll be able to draw a conclusion. So you find a problem or a research question, first of all. For you to approach is, for you to do a synthesis, it means that you must be able to collect data, especially primary data, experimental data that has been published, no matter whatever year you want to cover, depending on you. You can start from 1989, 1990. There are databases that could give you such information. So you can bring all this data together, articles together, and you begin to synthesize them. How do you synthesize them? You would have identified your research question, which is very critical. I'll be talking about that. He will be elaborating on that in his session, but I'll be touching on that. So from that research question, you'll be able to develop what are the inclusion criteria, what matters to you in each study, because at the end of the day, if you get into PubMed and you type in your research question you are looking at, maybe you want to determine why do we have much people having diabetes in South Africa. Some people might have published on that. So it gives you 2,000 papers. Wow. So out of the 2,000 papers, which one do you need? That's where you have included studies and pre-specified inclusion criteria. So from those 2,000 data you have collected, you need to check what are my included biomarkers I'm looking at, parameters I'm looking at, then you begin to synthesize them. All right, there are different kinds of research syntheses, and there are many. And if you look at that reference, you may want to go through, very critical. It touches on many, you can read, you may not understand all, but you can understand some specific to your field, all right? So you may want to go through that paper. It will give you more information about research synthesis. So we have the quantitative research and the qualitative research. The quantitative research much more deals about numbers. If you are in the biomedical research field or microbiology, or chemistry, food, biotechnology, etc., you will be familiar with quantitative research because you deal with numbers, you measure in terms of scale, numerical data. All right, so you express your data in numbers, all right? From your sample, how many effects, what level effect did you have? What level of activity? So you get a number either increasing or in a decreasing manner. That's quantitative, all right? And you have to do some statistical analysis to be able to ascertain that there are differences, all right? And you may have larger sample size most times in quantitative data. But for qualitative data, you must formulate your hypothesis. I didn't talk about hypothesis in terms of quantitative research. You are testing an hypothesis. So the hypothesis is already there. Okay, for example, it has been ascertained that continuous consumption of high levels of sugar may trigger diabetes. So you want to test what can mitigate hyperglycemia, high blood glucose in a particular patient. You want to test the hypothesis. But in qualitative research, you have to formulate your hypothesis, all right? You have to formulate what you want to do, okay? So you have to brainstorm, sit down. It might not have been done before. It might be from the conclusion of a particular paper, you're able to identify your hypothesis. Okay, somebody investigated and said, beetroot can mitigate hyperglycemia in a diabetic patient. And you check beetroot, okay? So what's in beetroot? You formulate your hypothesis. Is there something in beetroot that can reduce hyperglycemia, all right? So it's most times expressed in words. Most qualitative research you see are around narrative reviews. A narrative review is just like your literature review that you keep. It doesn't go in a systematic manner. And you decide what you want to write. You decide the conclusion and things like that. It's most times have a smaller sample size, and you are able to summarize, categorize, and interpret your data in terms of you want to determine in your school, your professors, are they doing well? Can you measure them satisfactorily from 1 to 5? Very satisfactory, excellent, bad, good, or poor. So from that, if it's a qualitative research, you can indicate 1 for satisfactory, 2 for bad, 4 for poor, 5 for excellent. From that, if 20 people pick 1, 30 people pick 5, you are able to analyze that statistically. But in qualitative research, you don't do that. What you are asking them may be, how satisfactory are they doing? Are they giving you notes? Are they attending to you? Do they keep to the appointment? So 10 people say they don't keep to their appointment. 50 people say we don't even see them at all. 10 people say they are excellent. They call us, we do Zoom meetings, virtual meetings, they help us in the lab. So you are now able to categorize them, okay? Lab activities, they are always there. Zoom virtual meetings, they attend to us. And you interpret the data. So that's the difference between qualitative and quantitative research. I need to move a bit faster now. So we have different forms of synthesis, and it could revolve around quantitative research and qualitative research. Some may be specific, but some may revolve around. But if you read that paper I talked about, you will be able to identify which is specific and all that, and whichever one that is specific to your field, all right? So we have the thematic synthesis, realist synthesis, meta-ethnography, meta-aggregation, and many others. In the quantitative research, we have meta-analysis, we also have meta-regression, diagnostic, meta.
Slides for those
Online. Oh, interesting. Okay, let me check why that is happening. I don't know why it's not moving actually. Okay, let me reshare mine. Okay, can you confirm online? Just send a message if you can see the slides. Let me just move it, and if they are moving now, please kindly send a message.
Are you moving again?
Okay, somebody said they can see now.
Well, if you move to the next.
Okay.
It's not moving.
All right. And now? Okay, all right. Okay, just send us a message if you are online, if there's any challenge, all right? So I will be narrowing down to 2 minutes. Getting feedback. Okay, sorry for that. I'll be narrowing down to meta-analysis in this presentation. But before I go into meta-analysis, let me just speak around some of the review approaches. I've mentioned narrative reviews. Most of the time, when what we're told is that most narrative reviews are written by experts. Most of the time, editors from journals identify experts and invite them to write narrative reviews. But you can also do so. Get into different databases, download papers. You identify your research question first, what you're targeting, all right? And many times there are no strict rules on how studies in a narrative review have to be selected. No rules attached to it. Nobody says you must stick to this, you must stick to that. You identify the themes you want to speak to. You identify the subtopics you want to address yourself. Except the reviewer says, okay, can you also address this? But you may decide to do so, and you may decide not to do so. And there are also no fixed rules on how to draw conclusions from the reviewed evidence. And this is why there is a limitation to narrative review, because you will always draw an evidence to suit what you want most of the time, maybe so that your paper can be accepted. But if there are other papers showing negative effects or activities, you may want to be silent on that. All right, that's one of the limitations, and that's why it leads to so many biases. This paper was my first review paper that I published in this journal in 2017. And one of the advantages is that it gives you a lot of citations when you publish narrative review, because it's maybe broad. So many people looking into that area may want to cite your paper. All right. Okay. Systematic review usually summarize evidence using clearly defined and transparent rules. And that's why it's different from narrative approach. This approach, there are rules to it which you must follow, and it's systematic. If you don't follow it, the editor, the reviewer will query your work and it won't be accepted. We will be talking about those rules, what you need to speak to, and all of that. Okay. Research questions are determined beforehand. That's one of the rules. You must be able to highlight your research question. What do you want to do? Okay. Some people have indicated that cannabinoids, which is from cannabis sativa, can reduce memory impairment. All right, can induce memory impairment rather. And you want to see the association between cannabinoids or cannabis sativa and cognitive dysfunction. So you get online, you search cognitive dysfunction and cannabis sativa, and it gives you 1,000 results or outcome. And you begin to check who streamlined into cannabis sativa. And if you are looking at cannabinoid, which is an important compound in cannabis sativa, it will be something different from cannabis sativa generally. So from there, you will be able to draw your research questions and see experimental data, primary data on that particular research question. So research question is very key. You can't get into systematic review without a research question. That must be your first point of call. All right, you must identify, what do I want to look at? Okay, for example, you are into, let's look at, okay, maybe microbiology or mycotoxins or food toxins, okay? And there are different kinds of toxins that induce different kinds of diseases, maybe diarrhea. So you want to look at one particular organism, the association between that organism and diarrhea. All right, what are the markers? The papers, the experimental data will tell you the markers you have to look at. All right, so that's very key. So it must be explicit, reproducible methodology. That is, I must be able to follow that methodology, all right, and get at least the same result you were able to get. You are not biased about it, okay? All right, next point. It aims to cover all evidence. Yes, it's going to cover all the evidence, primary data that has been published till 2021 December. So it covers all experimental data that has been done over the years. So you are able to deduce from the data you gather if it is true. Because if you gather 1,000 papers and your inclusion criteria, what you are looking out for, that a particular organism A can cause diarrhea, not all the 1,000 papers will report organism A, because all others will report only diarrhea. Maybe at the end of the day, 50 people, 50 articles, or 50 authors, all right, reported for organism A. All right, out of the 50, how many reported that this organism A caused it? Maybe 25. Maybe another 25 reported they couldn't determine if the organism caused it. So from there, you begin to have an idea. What are the markers that the first 25 authors looked at? What are the markers the other 25 looked at? So that's critical. So it tells you that it covers, all the evidence. They also access the validity of the evidence. That's part of the thing you're going to do, because you're going to access all the output you have to determine the evidence. All right. I don't know if you can see this. It looks a very bit it looks small a bit. This was a data from a systematic review. You could see different points, some of the study characteristics shows the authors, the country, the design, the gender, the age, the related diseases associated with what they are looking at, the dose used, all right, the control, and the endpoint. These were the criteria they set. You can choose to set, your own criteria depending on the study you're looking at, all right? And I want to show you this data synthesis from the study, all right, which makes it different from meta-analysis. I'm going into meta-analysis now, all right? If you read this, you will see that they said one investigator extracted key information from selected studies and 2 investigators. So it means that you can have many investigators. Here, we can all be investigators, okay? And technology makes it easy wherever you are. We can create a Google group, a Google form, a Google sheet, a Google Word document where you can populate and I can see it wherever I am real time, okay? All right, so they did it independently, and that is critical for systematic review. That if 5 people want to investigate a particular research question in systematic review, you have to go through the data. Mr. A goes through the data, Dr. B goes through the data, and I'm able to check what you are able to deduce to see if it's correct with what is in the papers without being biased, all right? So data extracted include country, sample size, participant characteristics. What that means is that in each paper, maybe you had 20 papers on a particular subject. You are able to identify from each paper the country where it was published, majorly the first author. One can be published in the US, another in South Africa, another in Germany, all right? What are the participant characteristics? Okay, this study looked at this effect in adults, age 40 to 60. This study looked at the effect on young people from age 10 to age 20. But you set an inclusion criteria because your studies is focusing on adults. So you pick that study that focused on adults. You exclude the study that focuses on children, all right? Okay, dosage and duration of the treatment was included. But look at this, meta-analysis or meta-regression were not performed because the qualitative assessment of heterogeneity of the RCTs indicated that it would be inappropriate to statistically combine the studies. RCT is talking about the random control trials, all right? So they've done the systematic analysis, but they were unable to move on into meta-analysis. Why? Because there was no heterogeneity of the data. You will understand this in the next session, what we mean by heterogeneity of the data, because without heterogeneity of your data, you can't do a meta-analysis. So they stopped at systematic review, and that's the difference between major difference between systematic review and meta-analysis, all right? If you have heterogeneity in your data, you can go ahead and analyze your data with statistical analysis. That gives you a meta-analytic review or synthesis. But here, you are not able to move on. So you are only able to report or synthesize your data using this inclusion criteria. So you will not be able to determine majorly the significant difference between a particular study and another because you were not able to perform a statistical analysis because of the non-heterogeneity of the data. That's very critical. So let's drive down into meta-analysis. Okay, all right, that's fine. Meta-analysis is an advanced form of systematic review. The scope of meta-analysis is clearly defined beforehand. It follows almost the same route, because if you can run a systematic review and you have heterogeneous data, it means you can move into a meta-analytic review. So you see that is interwoven. And that's why you see many studies. They will say, okay, association between high blood glucose or high blood pressure and manganese, as an example. All right, a systematic review and meta-analysis. But you see some studies, they tell you association between the risk of exposure to manganese and memory dysfunction, a systematic review. What that tells you is that they didn't analyze that result statistically, but they were able to maybe draw a conclusion by narrating or by concluding, by interpreting each data that manganese, which is a toxic, neurotoxic metal, can induce memory dysfunction. All right. And maybe from their result, they saw many papers that says low levels of manganese did not induce neurotoxicity or memory dysfunction. All right. Okay. So you must determine the scope of your method. Scope is very critical because most of the time you will have large data sets. Your research question may your keywords for your research questions may bring up to 1,000 papers, but your scope will determine how many papers you will need out of the 1,000 papers. That will be critical to your study. All right. Okay, and the same way for systematic review, it must be reproducible. All right, I've mentioned systematic analysis and meta-analysis review papers. Meta-analysis aim to combine results from previous studies in a quantitative way. Now, systematic review can be quantitative and can be qualitative, but meta-analysis is specifically quantitative because you must express it in numbers because you've done statistical analysis. But for systematic review, I can turn a systematic review into a narrative review along the line. I will touch that before I round up. But you can also do systematic review in a quantitative manner. All right. For example, you are looking at the effects. Sorry, I'm always talking about diabetes, memory dysfunction, and things like that. That's my research. I always have flair for that. Okay. You're looking at how reducing your blood sugar level can mitigate diabetes. And you find out that at the end of the day, you had 20 good studies that reported on that. Now, you can now be going to touch on each study, the data they got. Maybe their insulin levels were low, for 3 months out of 12 months in the year. You're already touching quantitative, expressing in numbers. But the same way you can do it qualitative and not mention the numbers. So that's systematic review. But when it comes to meta-analysis, there's no how you won't deal with numbers because you've done a statistical analysis, all right? Compared to systematic reviews, meta-analysis often have to be more exclusive concerning the kind of evidence that is summarized. Why? Because you've done a statistical analysis. So you can deduce and interpret your data in an exclusive form. To perform a meta-analysis, it is usually necessary that studies use the same design. And that is why heterogeneity comes in. If studies don't use the same design, what do I mean by that? you are looking at a research question that says, like I said earlier, compound or a particular organism A can induce diarrhea. All right, okay? But they used different methods in that particular paper. All right, they were able to identify the organism. They were able to do some molecular techniques. They were able to do maybe gene expression. They were able to do other metagenomic studies depending on where the samples are coming from. But not all the 20 studies will be able to run the same research design. A study can be done in maybe Monrovia, Somalia, where they don't have facilities to molecular techniques. So they are not identifying, they are not reporting molecular techniques in that study, but it is part of the 20 studies you have gathered. So in looking at that, you can't say that you have heterogeneity in terms of the same design, all right? So you must look at what are the studies that have the same design. Maybe out of the 20, at the end of the day, 5 used almost 7 the same methods, all right? 7 methods. So you're able to concentrate on that. That 7 possibly may give you much more heterogeneity, all right? So that's very critical. So look out most times for same design, same type of measurement, the biomarkers they are looking at, the parameters they are looking at, is it the same in the 10 studies or 10 articles you have identified? So it's critical for meta-analysis. So this is one of the paper we, okay, thank you for sensitizing me. I'll be running off shortly. One of the papers we published recently in scientific reports that speaks to Mycobacterium avium subspecies paratuberculosis related type 1 and type 2 diabetes mellitus. I was involved in this study, and I was part of the persons that collected the data, all right, looked at each manuscript. There were some studies that reported type 1 alone. They were excluded because we are looking at type 1 and type 2, all right? There were some that stayed on type 2 all alone, okay? But our studies covered type 1 and type 2. You may want to check online for the paper and read through. It may also guide you, okay? This is one of the tables from the paper. If you look at it, most likely you don't understand some of the things there, but those are things peculiar to meta analysis. From your statistical analysis, you will be able to get some data which you can't speak to. He will be speaking to that in the next session, and you'll get more clarity. So let's look at some critical points. I just have less than 28 more minutes, and I hope you are able to follow the presentation. If there's anything you want me to go back to, I'm happy to do that. Okay. For you to conduct a systematic review or investigation or meta-analytic review, there are some critical points you must master very well. Okay, specific to your field. I've mentioned research question. First of all, your research question gives you a scope. Your research question gives you what you want to investigate, what you want to cover. So I'm not going to dwell much on that again. Eligibility criteria, okay? Your eligibility criteria is what you use to identify what you're looking for. Like the previous paper, we were looking for type 1 and type 2 diabetes, Mycobacterium species that are able to induce type 1, type 2. Don't forget, Mycobacterium avium subspecies paratuberculosis. So it tells you that there are other Mycobacterium species. We are not looking at those ones. Most likely, When you get to PubMed, your research, search databases, Web of Science, Science Direct, Scopus, by the time you get keywords from your research questions and you put it into the databases, Mycobacterium species, avium, paratuberculosis, and diabetes, you are likely to find papers that talk about Mycobacterium species that never talked about diabetes. That's not part of your eligibility criteria. So you're excluding that paper. You are looking out for, you first of all look at the title, the abstract of the paper. Does it speak to Mycobacterium paratuberculosis avium species? Does it speak to type 1 and type 2? That is what you're looking for. And we'll be letting you know the search tools you can easily use. There are search tools, software that can easily help you to detect these things that, it makes it easier for you. We'll get there shortly. All right. The PICO framework is very critical. And when you talk about PICO framework, it talks about translating your research question into your eligibility criteria. And you need to use the PICO framework. If you read that paper, and some other ones I'm going to point out to you, you will understand this more. It talks about, one, the population. Okay, you are looking at tuberculosis, Mycobacterium tuberculosis in type 1 and type 2 diabetes. In what population? Adult population? Most times, most adults don't have type 1. So you need to set what age range are you looking at? Okay, what species, if you are not looking at human population, what species of organisms are you looking at? So that's very critical. Is it in young people? Okay, is it in adults? Okay, so intervention in this study, what kind of intervention studies was done? Was it a treatment study? Was it treated? All right, okay. And you're also looking at if there is a control group or comparison. Did they only use control in those papers? Was there any comparison? Maybe they use a control, the placebo. All right. Placebo is maybe the control they just give what cannot induce any disease, maybe like water or saline or things like that. All right. Or did they compare it with other sample treatment? For example, you are looking at acarbose, which is an anti-diabetic drug, association of acarbose and diabetes. Okay, as a treatment intervention. But if it's only speaking to diabetes alone, there is no intervention in that study. All right. So what about the outcome? Outcome is very critical. What did they deduce? Is it only the average of 10 subjects, 10 or the average of the population, which is the mean? Okay. Did they include standard deviation? That is the SDs. There are some statistical analysis done in those primary data. You need to pick the mean, excuse me, from each sample and the standard deviation because you will need it for your meta-analysis. All right. Was there any form of other data that is available that you are able to pick that is needed? That's very critical. We will look at that in the next slide. You need to have an analysis plan, okay? You need to do a study search. I've mentioned that you generate keywords from your research question. An analysis plan is just, how do we want to carry this analysis? You have a research questions and you have 10 people in the group. We need to define a methodology or a plan to be able to carry out this analysis. For example, when we wanted to investigate that particular study, each champion did, okay, brought up the research question, Dr. Kundayo now, and said, okay, Tosin, take these 10 papers and work with. Okay, there was another person in Cape Town, take these 5 papers and work with. That's an analysis plan, all right? And everybody brought the data. He went through the paper one after the other, okay? We exchanged papers as well to be sure that there was no bias, all right? That everything was done correctly. So you need to have an analysis plan. Also, you need to do a study search. That's when you get into your database. That's in the next session. You will be talking about that. So we'll know more about that in the next session. There are different databases available that you can use. You need to do a study selection. That is where you pick out of the outcomes from your study search, the studies you really need. You may be excited about other studies, but you don't need to pick, no matter how excited you are about them. This study is good, is interesting, but does it speak to your scope? Does it speak to your research question? So you don't need to bring emotions, or because you will do quality assessment at the end of the day. If you are biased, your reviewer will be able to pick that. And you don't even know, maybe the reviewer reviewing your paper is one of the author of the study you picked. So if you are biased and you are saying what he didn't say in his paper, you know the outcome, right? So you need to do data extraction. We'll be speaking to that in the afternoon session, how to extract data, all right? And we'll be doing some practical demonstrations on how to do that. Also, how to extract data and all of that, using different tools, using different applications is very key. You need to identify what data do I need to extract, because there will be many data in different studies. So you need to highlight, okay, I'm performing these studies. What are the parameters associated with this study? All right, those parameters are what you stick to. If you like any other parameter, you don't need to pick it. Pick what you want to speak to. That's very critical. And that is why there are rules set, and you must follow those rules. You will do quality assessment. Quality assessment in terms of, you are not the only one involved in the study. We have about 20 people in the group. So we have maybe 120 papers to work with, depending on the study. So I sent 10 papers to Dr. A, and maybe Dr. A was busy. We gave him 1 week deadline, and he was busy in the lab as a postdoc. He didn't have time to review those papers to extract the data. And he doesn't want to look back to the group that I must submit something. So on the last day, before 12 midnight, he begins to check first paper, blah, 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 okay, 10, 20, 50, puts it there. He didn't see other data to pick from. He's already biased. So out of 20, maybe he works with 5 and submitted the 5 results, complicating the work for the group again. So whoever is coordinating has to check, or you assign 3 people. Let's do qualitative assessment. You go through, out of the 20 that we sent out, go through 5, I'll go through 7, you go through another 8 that you didn't go through earlier. So you look at what they have done. Did they map it out correctly? Did they identify the parameters correctly? Is the mean actually 10? Is the standard deviation actually 4? Or he was sleeping and thought it is 14? So you have a mean of 10 plus minus 14. Are you getting what I'm saying? So your quality assessment is able to pick that, and many other things which you understand more. So the next thing is you do the synthesis, all right? You use the software, you input your data, you get your result. Then from that, you are able to see a deviation that they said organism A induced diarrhea. But out of the 10 papers you were able to analyze, there is no heterogeneity, or there is no standard deviation. Okay, all right, and you are able to interpret that. Yes, though some studies reported, but out of the studies we have gathered from 1989 to 2021, we can't vividly say that organism A is able to induce diarrhea out of all the 10 studies. I don't know if you're getting what I'm saying. So that helps you to come to a logical conclusion that can even form health policies in the region or the location where you are carrying out the study. Yes, out of 10 papers which you gathered, all right, one person reported that organism A induced the diarrhea, but 9 could not report that. So by the time you carry out your meta-analytic review, you are able to draw that conclusion. All right, I'm rounding up. Okay, you want to help me with my... all right, I need to run very fast. Applicable software. I will encourage you to check this paper. It talks about comparison between softwares, ones that are very good, disadvantages, advantages, and all that. And these are the different softwares you have: comprehensive meta-analysis to RevMan. RevMan is from about 10, 15. Okay, all right. Okay, I'm still online. Okay. All right, so these are the softwares. I was trying to tell you that RevMan is very, very fine from Cochrane. You may want to check Cochrane library. You will find useful information about systematic reviews and meta-analysis, all right? Okay. So this is it, Cochrane, RevMan, Review Manager. Check your institution also may have access where access is limited as well. All right, so I will touch on science mapping and bibliometric analysis before I round off this session. I hope you've been able to get some understanding around what we'll be talking about. So science mapping is a bibliometric tool to analyze and mine scientific output. Now, when I started my PhD, I always thought narrative review is what you can just do, all right? But he introduced me to bibliometric analysis. In fact, I think the first paper I published on bibliometric analysis was in +1. So I saw the paper and I was like, wow, interesting. So you can actually do this. And I became interested. Bibliometrics is an essential tool for assessing and analyzing academic research output, contributing to the progress of science in many different ways. In fact, you can't do bibliometric very easy because you can analyze research outputs for as much as 1990 or 1980 to 2021. You know, I published a paper recently, and one of the reviewers' comment was that, how can you say you are reviewing a paper from 90s? Why is in the database? Did I go to, you know? So that you are able to see the trend of research output on that particular research question from 1980 to 2022. You are able to see the research trends. Maybe there is increase in research output in diabetes, maybe from 1980, maybe to 1990, And from 1990 to 2001, there seems to be a drop. And from 2001 to 2022, that bibliometric analysis is able to give you those, outcome. I will look at that shortly. It provides objective criteria to access research developed by researchers, being increasingly valued as a tool for measuring scholarly quality and productivity. In fact, where this is very interesting is that it's able to measure the productivity in that area of research or that research question you formulated. For example, during his PhD, Dr. Kundayo's PhD was able to work on Plesiomonas, Shigeloides. And one of the things I think he discovered was that there was not much research output on that. I don't know, maybe that was what stemmed him to look at the bibliometric analysis. I'll show you that paper shortly. But you are able to see the quality and productivity. You are able to see different trends, different features in terms of that research question. All right. It describes the aspect related to data processing, analysis, options, and visualization. Yes, it shows you the graph. It shows you the table. How many authors have published that research question you are aiming at? The total citations of each author, the country from where those authors have published, you're able to see them, all right? And this paper I was referring to, a global bibliometric analysis of Plesiomonas related research. And if you look at the conclusion of this paper, I'm not sure if I pointed it out here, okay? One of the things he was able to point out to was that the research output in terms of this particular organism is very low. Why? Because he was able to run a bibliometric analysis from 1990 to 2017. But if you get on PubMed and you put in Plesiomonas shigeloides, you may find maybe some papers and it tells you maybe page 1, maybe to 20. You think that you have so many papers on that particular organism. But by the time you start checking, you just see that somewhere shigeloides was mentioned. But by the time you carry an analysis, you find out that the research output on that particular organism is very low. All right, there are some tools required for you to be able to do this. You have to do a search. I won't dwell on that because of time. He will be talking about that. Using Web of Science, Scopus, GSMA, and others. And to perform it, you need some software as well. Cytopy is very common and very accessible. And that's the common one I know, and is linked to Web of Science and Scopus as well. You just need to know the technical aspect of how to use this software, and you are able to generate your data. All right. Okay. And other softwares used to conduct this, BiblioShiny, BiblioMaps, Site2, the VOS viewer, will help you to view your outputs, your figures, your tables, your data, etc. And outcomes from this research, from bibliometric analysis, some of the things you are able to get is research output, like I said, country output. How many papers were published on this research question in China? It will tell you. It means that China is leading in that research. It means that US is leading in that research and is able to tell you authors collaboration, couple networks, that, okay, in this paper, you have collaboration between USA, China, and Egypt. In fact, I did a study, I was so amazed. And I saw that in terms of that research on polycyclic aromatic hydrocarbons, Nigeria had one of the top collaboration networks between US and China. So it means that there are some researchers in Nigeria collaborating with some researchers in the US and in China to get this work done. So you'll be able to have that information. All right. And this is the paper I was referring to, which just got accepted in Toxics, Neurotoxicity of Polycyclic Aromatic Hydrocarbons, a system mapping and review of neuropathological mechanisms. Why I want to point your attention to this paper is that I didn't limit this paper to bibliometric analysis. It was more of a mixture, bibliometric analysis sandwiched with systematic review. So you don't have to limit yourself, because you can send a bibliometric review paper to a particular journal. And in fact, one of the journals I sent the paper to, the first journal, they sent it back and said, this data is too much. You are just telling us stories, blah, blah, blah. So I said, okay, interesting. So I said, I can actually turn this into a systematic review. And I sat down, I started drawing pathways, started looking at the mechanism involved with what the bibliometric analysis, the outcome from the bibliometric analysis. So it revolves around it, except it's not you can't turn this into a meta-analytic review because you didn't follow the systematic plan, you didn't carry out the statistical analysis. All right, okay, but you can, find a way around to put it in a narrative review if you follow a systematic pathway to make it a systematic review. All right, okay. So in summary, we've talked about meta-analysis. We've talked about combining the result of previous studies in a quantitative manner. Don't forget that. It's not qualitative in nature. It is quantitative because you are talking in numbers. It synthesizes all available evidence pertaining to a research question and can be used for decision making. In fact, if you look at Cochrane is like not really a society, but a combination of researchers across the globe that come together to do systematic review and meta-analysis. Okay, it helps to make policies, the outcomes from systematic reviews and meta-analysis. To be able to form policies, to be able to form maybe some structure from that, depending on how you're able to interpret your data without being biased. Right? A good meta-analysis will be determined by a clear research question and eligibility criteria, writing an analysis plan, pre-registering the meta-analysis, and conducting the study research and data extraction in a systematic and reproducible way. Those are the things we have talked about. So I'm rounding off. So let's test your knowledge before I drop the mic. Some of the things you are able to get. So before I ask this question, so that you can revolve around that. Somebody wants to share what are the things, maybe something new you've learned you've not had before, or anything that has benefited you from what we have taught in the last 30 minutes. I just have 5 minutes to do this. Okay. Anybody wants to build the cat? All right. Okay.
You indicated that systematic analysis and a review can be a quantitative or qualitative type of study, whereas meta-analysis can only be quantitative.
Yes, that's correct. Yeah. Any other person want to say something? All right.
You also said that a systematic review does not contain data.
Okay.
But meta-analysis can contain systematic analysis completed with meta-analysis.
Okay. All right. That's correct. But it also contains data. Okay. Any other person wants to try? Okay, ma'am. Go ahead.
I think I also want to say something about research question. It's very important in meta-analysis because it will guide, it will give you a scope of what you want to do and also guide your steps towards achieving the research questions and make you focus also in achieving the aim of the review. Thank you.
All right. Okay. Thank you. We are not forgetting those that are online. So you may just want to raise your hand and I will recognize you. Okay. Please, if you have anything to say, just raise your hand. Thank you for that contribution, Sir Omar. Okay. You want to say something? Yes. Okay. You don't necessarily have to go to the lab. Okay, you can be wherever you are to do that. Okay, you want to say something?
For the narrative review,
Yeah.
the researcher has the opportunity to towards a specific topic, depending on interest. Whereas for meta-analysis, they are being guided by the available data and the information.
All right. Okay, that's correct as well. Okay. Any other contribution? Run off now.
One contribution.
Okay, please wait. I'll recognize you shortly. All right. Okay, ma'am. Go ahead.
My understanding from listening to
Hello?
we need
Go ahead, please. Go ahead.
we need more than one person to actually do the systematic review.
That's correct. That's correct. That's correct. Okay. Before we recognize those that are online, you want to say something? Okay, go ahead. Yes, that's correct. That's correct. That's correct. Okay, for those online, anyone wants to say anything, you can unmute your mic and go ahead. But please, can you raise your hand so we are able to do that in an appropriate manner? You can unmute your mic and speak. Anyone? Okay, I think there's no contribution from online any longer. Okay, on Zoom. Okay, lastly, anyone wants to answer any of this question? Okay.
You need to have your research question defined. So
That's correct.
Yeah, so that you can just include all those studies,
That's
take
correct.
all the parameters, and exclude the other studies.
Okay.
But you won't be able, you won't be biased
Okay.
in your study, and you won't be able to give wrong results to the readers.
That's correct. That's correct. So we've answered the questions. So,
Can I
all
also
right.
say something about, sorry, sir, can I say something about difference between systematic review and also meta-analysis?
Please go ahead. You can.
It borders on the homogeneity of the data.
That's
if
correct.
the data, yes, for meta-analysis, the data has to be homogeneous, sort of. But for systematic review, it may not necessarily be homogeneous. And then also the what are that things? I think that's the area that the difference I can spot out there, sir. Thank you.
All right. Thank you. Somebody's unmuting. Kunle, that's going to be the last person, and I'm going to round up. You want to say something? Okay. All right. Okay. So that brings us to the end of this session. Once again, I want to appreciate Prof. Thank you so much. Questions. We have a session for questions, so we don't want to, but you can just note them. Please write your question down. There's going to be a session where it's going to be an interactive session. Thank you once again.
Please, I want to add to the issue of homogeneity and heterogeneity.
Okay.
After you might have gathered your data, you might think they are homogeneous, whereas they are not. There are tests for you to determine maybe they are homogeneous or not. Because there are various reasons why the studies might not be homogeneous, despite the fact that they talk to the same topic. One of the reasons is the sample size, the sample setting, the sample location, the study population. So you might, but at the initial point, your data must be on the same topic, but for example, some might carry out his own experiment at room temperature. Some other person can carry his own experiment at controlled temperature. Then, mind you, you know that room temperature always varies. So those things will affect whether the outcome at the end of the day is homogeneous or heterogeneous. So you have to, at the end of the day, there are various tests to dig deep into it to know what causes the heterogeneity. Thank you.
Thank you so much.
Thank you so much, Dr. Tosin, for taking us through the introduction. And I'm sure a lot of us have known more about meta-analysis, systematic review, and science mapping. So now we
Sorry, there is no sound. We can't hear on the online session.
this.
Okay.
Any time you have to share which browser.
This one, Opera Mail.
Okay, which one now?
Let's use this one.
Yeah. All right. Okay, this is what I see. Operator. This is the tab, right?
Yes. Oh, don't share those. Yes, I want.
He's seeing it as this. He's not seeing it as this. What's happening?
Okay.
So that's a challenge. I think. Can we try this? That's a challenge. It's not seeing any of the Opera tabs. Okay. Okay.
Sir?
Yeah, it's not sharing this. I think this is. I open this in another page.
I have another.
Okay. Open it in maybe another browser.
Browser.
Yes.
We need to use.
Okay, let's use application window. Can you scroll down?
This one. This one.
Trying to share the current screen is working on, but it's not sharing. Yeah.
Yeah.
Yes, it's showing that it's sharing the screen, but it's not showing on the. Not showing on the screen. Okay. Yeah. All right.
Okay, fine.
Yeah.
Thank you.
Yeah, we can see online also.
Okay. Thank you. Let's Google search Scholar. Then the first link, Google Scholar. Let's click on it. Now we can see the search bar, but I want us to go to this 3-bar line so that we can. Am I too fast? So that we can access the advanced search. Now, before you do your advanced search, try to select the setting. Normally, the default is 10. So increase it to 20 so that you can have more view at a time. Then after that, save. Then we'll go back to that. We click on advanced search. Then this dialog box will come up. Now, we've defined our search term already. We have Shigella as the main one. Please, we have short time. So, okay. Now where I will assume that we have been able to set the output to 20, then we'll save it. After that, we go back to the 3 bar, the left, the upper. Then we click on advanced search. Then when we click on advanced search, this dialog box will come up. Now we have stated our what is it called? Our keywords already. So we are to type in our keywords now. The first thing is Shigella. Shigella. Then now you also want to search. We also want to search make, mix, making, made. Here we have it with at least one of these words. With at least one of these words. But if you want it to be exact, you put it here. When you don't want any option, you put it here. Then, so let's enter here because this stuff, I've already entered it before just to practice, so that just enter it without any comma. Make, mix, mix, making. Then we have where my word occurs. Do you want it anywhere in the article? Which one do we choose? Or in the title of the article? Why? Why do you think title of the article will be the best? Yes, in the title. If you put anywhere in the article, the work might only it might be the citation, the reference that talk on me, not necessarily on this particular work. You bring it out, then you have a load of work to do when you are removing duplicate. So we are going to use this. Then the next thing is hit the search icon. No, we want, now you specify the year when you have interest in a particular period of year. So now, this, you may need to record this 79 results. Result, copy Google Scholar search, result, 79 results. So now the best thing is, how do we get all this data downloaded? Now, you know, Google Scholar doesn't have a bulk of way to do. So the best way to do is to create a library within your library. So what you would do is to click on this star, which is the save button. Don't mind me, I have a lot of library. Some of the work I'm working on that are under review. So you have it like this. Now you click create button. I've already created my own, but I won't create it. Let me just demonstrate. Create button. Then you put a descriptive name into it. Aha. Then you can put Shigella as it all. So you can see now. So then you hit the done button. So myself, I have the library already. Now, what do we do with the library we have created? This is what you will now do. You click on each of this one. Then you scroll down to your library, click on it, done. It will save. Go to the next article, done. Then save. Now, because of the time schedule, I've already saved some of it so that we won't have much time to wait. So let me see the one I've not, okay, this one I've not saved it into the library. then save it in the library also. Then now you might not be able to do everything. Just note how to do it. So the next step is to show you how to download from I want to be sure if when you are done with this page, all this page, you move to the next page until you save all the data, all the article in your library, the library you created. So the same thing. Let me run to the next. Now we can notice something. All this article. Who can tell me what happened here? Why are they not having any link? Now, what happened is in this place, we specify include citation. So some of the work cited in those work that speak to our study will also be included. But if you don't want them, you untick this one to remove it. Now, after we are done with this, then you open your library. My library. Go to my library at the right hand. Now, don't mind me, I have a lot of library. So you scroll down to library for this work, then you open it. Now, you now have option export all. Export all. So another thing before we export the data is that you can create alerts. Let's go back to so that you monitor new input right from the day you start working on the title. So here you can create alerts. Just click on it. Specify your email address where you want the alert to be sent to every time. Then click on create alert. So I've not created alert for this one. Then create for. So I've created alert. So now let's go back to the library. So with this, even your research area, you can monitor new papers. When it's coming up, you can, okay, this work has been done, you look at it, you can easily follow on new development in your feed. So now let's go back to, make sure you are in the library. Now we export all. Now I told you, depend on the reference manager you want to use, you specify here. I'm using EndNote because we want to use EndNote to remove duplicate. I will click EndNote. The RefMan, if you are using RefMan, you can use it. You can use some software allow CSV. Then some also allow big text format. So make sure you click on export all article with the label. Now we'll download it. Then another thing is you need to create a directory or a folder where you will save this work so that you, let's create a library. Am I too fast? Am I too fast? So where are we lost? Oh, from exports. Okay, let's. Okay, let me go back to the pointer. I'm into my library. Now, sorry, I think I'm in the wrong place. Now, if you are just creating it for the first time, you won't have a lot of lists like this. Now, if you click on mine, Shigella is down. Yours might not be there if you have some library. Ma?
I think one of the challenges is that people must have a Gmail account.
Yes.
If they don't have a Gmail account, then they cannot be able to do what they're doing.
Yes, thank you, ma. Yes, it's correct. Ma. Yeah. Do we all have Gmail account? Okay. Okay, just follow and understand it. When you create your Gmail account, you can always follow. So now, export all, we have different formats in which I'm choosing EndNote because I want to use EndNote for the work. If you are using what is it called? Mendeley, you can use big text. If you are using Sotero, I think Sotero we use Redman. So you must know if you are using R package for it, you use big text. But there is no package in R that can handle data from Google Scholar. So that is why you can't use Google, R with Google data. So once you click on this, then you select export article on this page. You will have articles that are up to 200 in your library. If you click on this page, it will always return 20 because we have increased the space to 20. So it won't cover your article. So you need to click on export all article within this label. Then once you click on it, you export. I've exported it already. I don't want to duplicate it. So the next thing is create a directory on your machine or your PC. Where you put all the data. So the best thing is, normally I usually create my stuff within the download directories, or you might decide to use a different directory. So let me use a different directory within a folder I've created for this. So I will just say make, we know how to create folders. So Shigella, use descriptive names so that you can always relate with it. Now I've created it, I'm opening it, then from because I think I'm in it now, you can copy or drag into the new directory. So whatever, any article we are getting now, we start saving them here. So we've demonstrated how to use to export data from Google Scholar. No matter the topic you want to do, is the same approach. Then in the case of your search term, can be complicated when you have too many to handle. But if you use the search, the advanced tool, and follow the way we describe, it's easy to go through. Myself, I don't use this method, but I know it's easier. I can easily type right from here where I want to do in form of a command. I can say, because this one is more specific, I can remove some things. For example, I can say all in title because I want it to search the titles. Then colon, I will say Shigella, the first one make. Then in this place now, you need to use Boolean or mix. Your Booleans must be written in capital, mixed or making. Then the advantage here, I can, anything that I don't want, I can remove it by specifying minus review. Review minus case report. Well, please just stick to the to the simplest one for now. By the time you start interacting with these databases, you'll be able to do some specific search. So if I hit the search button now, we might have different results. Okay, now the main reason is my report is wrong. Case report. Or case, let me use case, anything case report to eliminate it. Then another thing is we might not have review articles. If there is no review articles, so those can appear. So now we've removed the review out of it, any review. So if you go to this, you know you have a tab for review. If you click on it, you see 0. Then if you say anyone, which is our primary article, so you can play around it. So let's also go into PubMed. We are starting with the free one so that if you are in a state that you don't have access to data subscription-based databases, you can still deal with do your meta-analysis and so let's Google search PubMed. In this case, we are trying PubMed now. The first link, just click on it. Then we have advanced search. Why I want us to use advanced search, we can also take control about what we want to see. So, mind you, the advanced search have user manual, which I want us to look at it at the end of the day, because it will tell you the different tag, what to do. It's just that I stopped. Okay, I'm not sure. Maybe people online can see the. I don't want to disrupt the stuff, but for people here now, these are just the. So from the this is the user manual, I guess. So these are the feed. Most of them that are important, and this one that I underlined in green, affiliation. For example, now I want to carry out a study restricted to African alone. So I will need to use this tag in my search to limit it to Africa. If you do it normally, you will have from other countries of the world, and you have difficulty.
We're not seeing.
Yes, I'm not displaying this online. I don't want to disrupt this stuff. I'm sorry. Then if you have interest in language, you have to maybe English, Spanish, Mandarin, Korea, whatever. You use this in your search. Then publication dates. This one is good when you want to do updates on previous search. Then publication type. In this case, the publication type can be systematic review, meta-analysis, original article. So you then this tag title, then this one is for title and abstract search. So you can always go through it. There are a lot of control which make it better. So let's go back to the main search button. So you can also learn from the interface by just looking at the available stuff here. So let's say now I won't be, okay, let's undo it manually. So the title, we are dealing with title and let's use title and abstract here. No, you type Shigella here. Then we have the add button here, add. Then the next thing is we add make. Then select or. Can we see it now? Then we have making, mix. Still leave it here. Then it's building the search term for us. Mix. Or making. Then hit the search button. So you can see the results. Now, there is a bit of error in the, let's reduce this to title. We have too many. Let's reduce Shigella to title. You shouldn't have to this. I think it's less than. So we still have, I think I'm not used to that one. Let me use my shortcut. That is a wrong, the search is actually wrong. The generating the stuff. So just try to learn read the user manual and look at how you can control. So I want to take control of the work now by writing the code to search. Now I will have Shigella because I want it to be title and abstract. You can short it TIM. I'm saying title and abstract. And okay, I think the search was correct. I think I made a mistake not putting and first. So now, because we want to nest a lot of and or, then you create a parenthesis in which you put all your. Now we have make. Then the search tab or mix. There is another shortcut to all this by listening to this. So I'll see demonstrate it. Then we'll compare the result. Making. I bit on my slide. I didn't talk about it. I'm sorry. We will be sending the slide at the end of it. So now with this, we can have specific results. It should be less than 70. Okay, 250, which is good. Now we've not removed reviews. We have not removed other form of publication type. So you can either do it, the first thing is look at it so that what you want to do, you are not duplicating effort. That is why I like using, what is it called? PubMed to do this. So you can, if you check review, how many review on it, and you can scan through. You can scan through moving antibody. You can, when you scan through the results, you will discover that none of them is trying to address what we want to address. Then you can continue with your work. So then if you actually want to know if there are meta-analysis on it, you can click the meta-analysis button. Mind you, some people will publish their own as research article. So you may not be able to get it. So what we will do now, we'll put into the search button to remove likely presence of meta-analysis or so not okay. Let's put it in the parentheses. Not review. Yeah. Yes, in capital. Then publication type, just short it as PT. Then or meta analysis. PT as well in the square bracket. Then if we hit the search button, we have less than what we had before. Now we are 240. Okay. Now, if you want to make it, you want to trade off sensitivity for specificity, all we need to change it to change this the tag here into title. So you will see that we have reduced number. We now have 63. But the main disadvantage here is we are trading off sensitivity for specificity. So, but for this, let's work with minimum number of articles so that we can demonstrate, because I want us to remove duplicate as well. Try to extract data from it. Then at the end of the day, we can expand it in a more better way. And maybe publish this as a group to mark this workshop. So now we've already have it. Then how do I get out my article? You don't need to, unlike, what is it called? Google Scholar, that you do it one by one. You don't need to do it. All you need is to send to, click on send to.
Before you go, on the search, that's the last one.
Are we okay now? So if you want to save the articles now, this you need to record this one. You record the results in a place. Then when you want to get the data, just click on send citation manager. Use citation manager. Then all results on this page. No, choose all results. Then create file. Hope I'm not too fast. We have short time. We have short time. Okay, create. Now, it's because I'm going to use PubMed, EndNote. That is why I have to download in this format. But if you are the one using R, you have to do it in another way. You use this save button, click save, select all results. Then you save in PubMed format. Then you create the file. So I will be able to use this So let's also get our downloaded data into the working directory. The next thing is, let's Google search Scopus. Okay.
I find it a bit easier to manage your articles or whatever you have done with it if you have an account. If you have an account, over there you are able to log in. You are also able to save all your searches. Not only that, you are able to follow up with a number of articles or the results. And if you are unable to, for whatever reason, if you are maybe in a hurry, you cannot really document your results, then you can save all of those and go back to it. Then all the search terms, you remember that when you are writing a systematic review, you have to include your search terms.
Yes.
You might have forgotten when you are doing this. If you have an account, you can always go back to it and get all your search terms. So I would suggest that people have an account with PubMed so that they can always go back to all of these searches and be able to access those data.
Thank you, ma. That is the best way. Myself, I think I lost my account with them. I wasn't able. But if you don't have what is it called? Account with them on your PC, you can always check the search history. Yeah. But the best thing is to have an account, create alerts so that you can monitor. But because I find out that Google Scholar is easy to give better update than PubMed. That is why I use PubMed. Most journals may not be indexed in PubMed. So, and mind you, you also have access to great research in Google Scholar, unlike PubMed and other databases. So that's why I usually use Google Scholar to track articles. But some of the articles that will be later indexed in PubMed will first come into Google Scholar.
And if you also have a seed account, PubMed has been able to link both accounts together. So if you lose it on PubMed, you can always access it via OSIT or CID.
Okay, man, thank you. We have a question from online, whether the search terms are codes or case sensitive. They are not case sensitive, except you are using booleans. The and the or not. All these booleans must be in capital letter. So please let's take note of it. Your keywords, they are not case sensitive, except the booleans you want to use, the operators, the and, or, not. Then depending on database, some database will say and, not, whatever the stuff. All those booleans must be in capital letter. So let's try Scopus. I think people here should have access to Scopus through the library, DUT library. Google search Scopus and look at checking access. Check your access. I'm using DUT University Library. For some of us that don't have access, let's just follow the same trick. Now, are we on this page?
We have on DUT have yes. If you're on school network, it's easy to get. If you don't have remote access, you may not have remote access. If you don't have remote access, you may need to consult the librarian, university librarian, to give you what is it called? Remote access. So let's just follow the demonstration. What's wrong? So it will bring you up to this stage in which you punch in your school email address and your login detail. Are we following? Please, I need a response.
Yes.
So we can continue. So if you come to this page, you will see different document search, and there is also advanced search that you can also exploit. This is the button for advanced search. Let's stick to basic search. Now, when you drop down this menu, you can see all fields, article, abstract, keyword, first author, article, title, then you can see affiliation, country. So if you have interest in country-related stuff, you can use those. You can specify it. So in this case, let's use title, article title for Shigella. Okay, sorry, I failed to demonstrate something here. Instead of listing all these making, you can use a shortcut by truncating them. So let me demonstrate it. I'm sorry, I just remember that I didn't explain. So we'll get similar results. When you use this with card, anything related to make will be returned in your search term. Either making, whatever, you will bring it up. So the results will be similar to what we had, 63 the other time. So instead of having a list of keywords, we have been able to streamline it into short one. So the same thing, you can do that on Scopus. So now we are putting Shigella here. Then you can add another field. And because, but in this case now, just like we have in PubMed, we want to search the article, abstract, and keyword. We just put, I think, let's just use the shortcuts that I introduced, just make, instead of listing all of them. So we can punch in the search button. Now we have 85 documents. So mind you, we have not removed the reviews. So here you can take control. We have the subject area. If you have interest in a particular subject area, the year, the type of documents. When you click down the the arrow, you can see article, book chapter, conference paper, notes. So for us, we don't have interest in conference paper or book chapter or notes. So we click on articles, then you limit your search to that. If you want to exclude this, you click on exclude. If you want to limit it to it, why this? Okay. So you limit it to this. Then suppose you have interest in region, country. You can always select the country of interest. For example, now if we want to limit our work to Africa, we just open all the countries available. And look, maybe we have Africa there. You know Africa countries by name. Then you select them. Okay, Ethiopia is the first. Wow, easy. So you are dealing with something relevant to our region. We get it. So that is how you know that you have a Nobel topic. So that means we have been sleeping. We don't take care of this part. And we eat a lot of milk and milk product.
Yeah.
Yes.
Yeah.
No. Okay. Please just focus on it. You will get something.
You know, I said when we started that we're not going to grab everything. At least you're just getting the basics.
This memory will help you when you are doing your own. You won't believe that I taught myself all this. When I was to pay for a training, I think it was 700 euros. So we need to appreciate Prof for providing this platform and NRF for sponsoring this workshop. So now you can use, if you have interest in language, we have language. So if you want to look at paper and Chinese, so in this case now, we are including all of them. We are not separating any form. But when most of them we have English abstracts. But if they don't have English if they don't have English abstracts, you can use Google Translate to add the idea. So I've used Google Translate in some of my work, and it worked fine. So there is no one publishing our African language. What are we doing? So we should be publishing in Xhosa, Zulu, and Swahili. Swahili is more. There are a lot of countries speak Swahili in Africa, so we can start. No, Google Translate can translate any language. So okay, let's get to download it now. Now at this point we select all. Then we export. There is an export button. Now you need to click all the fields because they are essential. Abstract, funding, and so if you are using EndNote, you use this. If you are using R, you can use these two. For science mapping, you can use CSV and any format that your reference manager will access. So let's export all the results. Mind you, you need to record all this. You must record in step, one by one. because they will form part of your methodology when you are writing. So you need to, so we won't demonstrate because of time. I think okay, let me just say quote something. Don't mind this. I'm using a remote asset from my alma mater, and I have to come here so that we have it on GUT. Just that I prefer this. There is a lot of similarity between Web of Science and Scopus. So we work in similar way. So let's just mention the code directly, not necessarily using the. Okay, let's follow the normal stuff. I'm just looking at time. Shigella. Then we add another row, and let's select. In this case, it's topic. I told you that topic deal with title, abstract, and keywords. So let's use the short keyword, which is make.
Just where Zinke was saying 433.
Instead of listing all the
Yes, thanks.
Now, in this case, we have 65. Now, you can see that the more Web of Science is stricter in the indexing, not all journals. So if you need quality papers, I'm not doing commercial for them. So just like Scopus, you know the paper here and the one you need. You can also record it. There are university. If you want to study how your university is performing in certain research area, you can look at the affiliation. You can check. Just then you want to check countries. You can also look at it. Now regions and country. So you can also look at it and select which one you want to study. So let's go up, and I think I've selected. Let's select article now. Article alone. Then refine to article. Now. Then we have 60 in this case. So you add to mark list. In this case, the page usually, I think the maximum is 200. So because you have some study that you have a lot of people up to 5,000. So all you need is to add them to mark list, record from 1 to the total number, which is 60. Then add it. The mark list will be highlighted here. You can zoom into it before we download it, or you can download it right from export here. Because we are using EndNote desktop. You click EndNote. You can also explore other net file. So when you get to this point, you also select record from this. It might not be, usually give you 500. You can You can only export 500 at a time. If you have more than 500, you have to do it in batches. So now I want you to record all the full record. Select this, then export button. So we have it now. So let's move this last two into our working directory. So we have all our data from all these three databases. So the next thing is to create an EndNote. Let's open our EndNote if you have EndNotes. Now, this is an ongoing work, so I want to create new one. That is the advantage with EndNotes. Please, are people online? Can they see the EndNotes?
No.
Dr. Tosin, please come. Please come. Let's and we'll stop sharing and share again.
Okay, yeah. Yes. Speech or application window. Yeah.
Is it up? I can't see it.
Which one?
EndNote, this.
Okay, no.
Okay.
No.
Okay, it's now within.
Come to end. Okay, go back here. I did not open it.
It's open now. This is
It's open now. Okay, share.
No, it's online. One people, I can see it now. Can people online see this? Please confirm.
Yeah.
Okay, fine. Thank you. Okay, the next thing is to create a new library. This is another work. I don't want to model it. This is the advantage of EndNote. You can create individual library for each project. So we create a new project, or you can just control new. Sorry, no. I can use to shortcuts. Why is it not? Okay, library. Okay, now you can navigate to your working directory and have it there. Inside the working directory, that's called name it, the new EndNote library. With Shigella Mic. Okay. So then close the previous, I want to close the previous library so that okay, now we can export our data into it online. Can you see it online?
Yes.
Okay. Now we can, the easiest way is to double click on the file. You will get into the open EndNote. So let's double click. You will see it being importing. So be careful that you are not sending the same data twice. Okay, the number will increase. Okay. Now we have all the data. We can remove the duplicate now from I think library. No, sorry. Yes, library. Find duplicate. Okay, no, sorry. All references. You can highlight them. Library. Find duplicates. So now myself, I don't like this. I want something that is automated a bit. Also, so now what we'll be doing is that we'll be exporting all these people to what is it called? To Microsoft Excel. There we can do a little bit of if you do it like this, you can. I think I made a mistake deleting what I needed sometimes. So if you just like it has been highlighted, we drag this to trash button. We remove the duplicate, but not all the duplicates has been removed. So you can as well also delete some of the articles that are not duplicate without duplicate. So the main thing is to use something that is a bit I want to remove all this article from trash and then back to okay, then all references now. Then we export it out. Well, before you can do it, before you can export it, you need to define the tab that you want to. So we create a kind of output style. You create new. Now in this case, now when you want to do duplicating, when you want to do duplicating, you can easily use okay, fine. What we need at this stage is the title, abstract, and the author's name, the title and abstract. So just choose type. Am I in the right place? Okay, author. Then tab, title somewhere down. Then create a tab. Then you look for abstract. I think abstract is down. Sorry, if you see it, you can guide me. The abstract. And which is the abstract, please? No, you need to pick it from the. Okay. Oh. Can they see it, please?
Yeah, it's fine.
Well, I'm looking for abstracts. I don't know what happened. Maybe I picked the wrong template. No, let's change the temp. No, that's the right template. Okay. Okay, so it's just to create. I don't know why we can't see the. Okay, it's down. Sorry. Now you save this as a new style. Sorry, I changed my. I think I put it in the wrong. Then save. Come put it, let's say Shigella as our style. So then you can close the. Now you just highlight all this, then export. You will now choose that style we just is the same as this, but you may need to select it before you can use it on the end. Don't worry, this can look looking as if it's taking our time, but when you are dealing with most people, EndNote is not advisable. So what we are doing is it's okay. Open Style Manager. Then we select the new one we just save as part of the ones that will be Shujela. Anyone seen it? Sorry, don't mind. Okay. Okay, we will select it. So we can close it now. Do we need to save? No. Then export now. Now we can see Shujela style. So all you need to navigate to the working directory and save. Where do we have it? Desktop. Save. So we are done with EndNote. The next thing is to use we can open our Microsoft Excel. Are we? Once the app is up. Thank you. Okay, now go to the working directory. You can browse to this place, but I don't like it. I told you I like shortcuts. Just copy the path link. I've already copied it. Now we go to Microsoft. Under data, select external, get data from external. Test file from test, because we are choosing test file. You need to browse to it if you don't copy the path. So myself, I've already copied the path. I'll just open directly. Now, just click next, and finish. You ask you where do you want it to start. Click it. Now we have the author, the title, and the abstract. So some people may not have abstract. So the first thing to do is, in some cases, you what did I have? What have I done? So I don't know what I did. Sometimes you can add spaces around it in the export stuff. What you need to do is highlight it, go to data, and then sort. Choose the column B, which is the title, and then so it will eliminate the empty spaces in between the exports. So now what we need to do is we need to remove the special characters, most especially publication in different language are usually have brackets in their title. So what we need to do is to remove such special character. And how do you do that? Just click Control H to bring it this dialog box. Then replace all. Since we are not replacing with anything, we will leave the replace with option free. Then the next, the other bracket, then we replace it with nothing. Then you can also look at it. Sometimes you can have special character, most especially on the language of origin. So if you have it, then you find a way to remove them. You can have spaces and so. Okay, now just select them. Go back to, okay, we'll remove. There is a button, remove duplicates. Can we see it? So let's click on it. It will ask you which field you want to use to remove duplicates. Normally, I use it, the title field, because the title usually different. You can have the same author having more than one paper in that area, and you will remove it if you use author or whatever. So now let's click the click button. Now it's telling us that 120 duplicate value found and removed. Then we have 164 unique value remain. So in EndNote, you need to look at this 120, all these duplicates one by one before you decide on which one you want to keep and the one you want to eliminate if you don't want to lose them. So what will take us several times is what we have achieved within a click of button. So sometimes because the indexing varies, there can be white space and whatever, you can still have some duplicates that you need to manage. So to do that, the articles are already in alphabetical order. You just follow them, any double entry, and you delete one out of it. So from one down, as I'm skimming through it, I have not seen duplicates. Please, let's follow. If we see anyone, let's alert me. $15 and $16. Yes, you are right. Yes, you can delete this out. 25, 26. Then another thing is also look at it. Which one? 27, 28. No, they are different. 26, 27. Okay. Now you must be careful. You know, we need the abstract. Some of them we have abstract, some of the duplicates we don't have. So retain the one that have the abstract, because we still want to use it to evaluate it. But in this case, the two of them, because they don't have the abstract. So I wish, okay, yes, they are the same. Yes, if you look at the author, they are the same. They are the same. So we delete it. So let's look at it. Okay, yes. $32, $33. So let's delete this, the capitalized one. Okay. Okay, this one, I think this one is the same. You can see why we don't have, the main reason why we are not able to remove the automatical is because, and then we have Russia. So let's just delete this. Then this one is the same. Then if you are the lazy type like me, rather than deleting, you can come to the next tab. You know, we have ABC, just assign line number, then sort it by the number. So for example, if I add duplicate, I can assign two to all of them. Then I write and sort by it, then delete at once. So, okay, let's follow this. Okay, let's follow this. We have these two, 49, 50. Are getting to the, then if you are the type 47 or 48, yes, you can this, if it's a kind of large number, you can begin to set some points. Okay, I have not saved it before. Let me save it. Differential efficacy of bovine. I think this one the same. Yes, we delete the one without abstract. $60, $61, which, okay. Sorry, we are taking longer time. Now, if you look at this, okay, they're not the same. In vitro. 57. 67. They're not the same. They're not the same. Okay, they are. They are.
$77 and $78.
77, 78. Yes. 84, 85. No, you should be careful. We have vaccine 1. Okay, they are the same. They should be the same. Those must difference because we can have vaccine 1, vaccine 2 written by the same author. Yes. So this is how we go through it one by one. And now it's because we have time constraint. If I'm dealing with larger data, I also take time to remove those special characters and whatever. Then be able to reduce the number, the minimum that you can handle within a short time. Because you can have up to 5,000 in some study. This one, okay, they're the same. How far? So timeline activation of this milk produce. $138. Yeah. So let's assume, because I'm not sure that we've all removed all the duplicates now. Let's assume these are the unique ones. So you will record it and minus it from all the sum from which you get from all databases as your duplicates that you remove. So now, the next thing is to start your review. Now, when we look the first paper, let's look at the title. Detection on Staphylococcus aureus Shigella in full sample. The title doesn't speak to it, then we need the abstract to judge that one. So let's look at the abstract. So I think I'm not going to read aloud. Let's just follow it. So we can see from this point now that the article, there is no milk as the subject here. Now, because we use, what is it called? Topical specific. So this one doesn't speak to it. So we remove it. Just like I said the other time, just assign, let's say 7. So we are assigning 7 to those ones that doesn't speak to the topic. Now, if you look at this one, merely from the title, we have milk and Shigella. We assign 1. The next one, acidophilic milk and carrier Shigella. Right from this, we have the title speaking to it. Then experience using direct in milk market. So milk is involved. Once you have what you want in the title, you don't need to look at the abstract at this stage. experimental study of the immune containing. This is related to me, but might not speak to our study. So for now, let's retain it, and we can deal with that during the mechanism origination of MIG-1 outbreak caused by Shigella. So now, in this case, this outbreak, we can only know when we read the article, maybe they sample MIG directly. If they didn't sample MIG, then we exclude it from our work. But at this stage, we still retain it. So I think I have 3 minutes to stop. This is how to do it all through. So now let's just, I will assign random number so that I will show you how to sort the one that you need. When you don't need. Now we have one, let's say we have more 77777111177. So make sure you, okay, let's just assume that this is the one that we want to sort. Okay, I think I can still. It's just to demonstrate something. This. At the end of the day, we have reviewed the title and abstract, and we have selected the articles of interest, and the ones that are not of interest we've excluded. So now we can remove those ones that we don't need of it. Using the sort button again, then the field is 9, is D. You can use you can name the field, then you can say sort in order from smallest to the largest. So now the implication is that all these ones are the study that we have identified at this level that are eligible. So these other ones, you might delete them out, or you leave it like this. Then you can copy the one of interest to next tab. Let's do that. But we have not done the actual total review. So we can copy it. Okay, the next thing now is to download all this article into the folder and start looking at it, extracting the data. So that's why some journals publish it as research article. You are doing a lot of work. Most especially when you have big data, like when you want to even send reviews to Lancet, all these, maybe Lancet or Ecology. I think the impact factor is something around 120, whatever. If you have Nobel title and you do meta-analysis, you send it to them, they will accept it. And they specify that you submit this as original article. So at this point, I don't know, maybe any one of us have any question.
I think we can continue after lunch.
Okay, sir.
Yeah. We can ask the questions later. But
okay.
But like you said, it's a lot of work. Ready, intense work. So.
Okay. I think we, Prof.
Yeah. Thanks so much. Thank you, Doc, for your presentation. And we have known more about analysis now. I think from after the launch, we're going to continue. So the launch is going to be from 1 to 2. So we can go for lunch. And those joining us online, maybe you can also have your own lunch wherever you are. So we're going to meet. We are going to meet at 2. Thank you. 2 South African time.
Okay, welcome back everyone to the afternoon session, both online on Zoom and here virtually at the venue. Let's settle down. We need to run quite fast. I'm sure we're able to learn a lot of things in the morning, first session, second session, and then we're able to pick some points. Time will not permit me for us to just have some interactive session on some take-home points on some of the things we've touched, but we've looked at basic introduction into systematic review and meta-analysis. And then we've also touched on looking at different databases, Google Scholar, PubMed, Web of Science, and Scopus. So I'm changing gear now and going more into the real work now, right? Which is where you need to get your data, analyze your data statistically. So I will be taking the first part of data extraction, and he will take the lead to how to analyze the data using the appropriate software. Understand that the rule is the same. All right. The rule is the same, but there are different approaches to it, depending on the tool, the software you're using. So quickly, I'll just run through data extraction. So I'd like to recommend this book. Very nice. I've read the first chapter. We want to look at it and doing meta-analysis. It talks about everything we've talked about: systematic review, data extraction, everything. Very simple, easy to follow, and easy to grab. So if you want to get it, it's online. You want to download it, all right? So I'll be taking you through how to extract your data, what to do, the outcomes you can look out for, depending on the study you're looking at, all right? There are different kinds of study. You have clinical trials, randomized control trials, observational studies, case reports, depending on what your target is. So where to find data in the articles, we'll talk about that. And we'll look at extracting data using different software. You may get to a point where you need to extract data from a graph, bar charts, line graphs, histograms. We'll try to touch on that. I'll finalize that, and Dr. Kundaya will come up to continue. So let's get into data extraction. It involves extraction of data, looking for relevant information that you need. Like I said, there will be several kinds of information, but always target the information you need. Okay? Specifically the information that answers your research question. And if you're looking at meta-analysis, it's about retrieving primary data And you will see that we're excluding reviews, case reports, because we want to target experimental data where you can get your primary data. And then depends on different kind of data sets available, different kind of samples used. It could be human trials. So there will be different kind of samples, maybe from 10 groups or from 20 groups who have to extract such data. So it also covers study characteristics, descriptive information, and the outcome data. I'll be narrowing down into the outcome data. So three major information to extract. I got this from that book, and it was quite explicit. Is that the characteristics of the study? You need to watch out for that. The features of the study was parameters, an observational study is a higher, depending on which if it's rats, animals, whatever. Then data needed to calculate effect sizes. You'll hear more about effect sizes in the next session, so you understand more. Study quality or risk of bias characteristics. This is very important. Your study quality is what your reviewer wants to see. If it doesn't meet up with the quality, depending on the rules set, you find yourself going back. One thing that is challenging about it is that you don't want to go back to that Excel kind of a thing. Duplicate, going back to study search again and to start all over again. So your study quality is very critical. All right. So data extraction, first thing I was able to touch on is plan your data extraction using your own methods. Okay. I was watching the study from Cochrane, what they did on systematic review. And it amazes me how they are able to organize a form where they listed what data they want to extract. And some group, they had to, they brought up a Google form so they can share it among the groups, and they listed what they want to extract. So you go to the Google form, as a member of the group, you fill it depending on the articles you are given. So that's a plan. You can decide to print the 10 papers and begin to search for the data manually. But it can be tedious when you get to a place where you need to extract the data from a graph. So the question is, if you have a bar and you have your error bars, do you want to start using ruler to measure the SD if there's no table that shows you plus, minus, so and so? So how do we do that? I'll be narrowing on that and finalize there. So avoid extracting all data. This is going to be very cumbersome. You can't extract all data. Be specific. What are the data? There's no rule that says that you must target a research design that has 10 parameters. There's no rule that says that. You can narrow down on 4 parameters. Okay, we want to look at the study that conducted qPCR on this study, the study that did HPLC metabolomics, maybe they did immunochemistry, or any kind of technique, depending on what you are looking at. All right, you can narrow down on 3 up there and look for 10 good articles that has that kind of method and analyze it. It's going to be fine. So develop a guide for data extraction based on your review team. Sit down with your team, develop a guide, discuss it. This is what we want to narrow down to. Let's stick to this, and you extract what you need to extract. It's also key to check and cross-check. So you have 10 papers, You have 5 persons on the team, you distribute into 2-2. After everyone has submitted, redistribute and let everyone check again to confirm it follows all the rules, the guidelines, and all of that. There will be general data for you to extract in terms of information that will be applicable to everything you want to do and what your reviewer wants to see, the readers of your article wants to see. For example, you can't show us a data without indicating the author's name. The author's name must be on the table, right? For example, the last paper we did together, where we had to settle how we're going to organize it. We agreed that I like to do things his own way when we have to work, because we tend to send things easy. And I like to find easy way to also do it as well. So I don't give myself headache. All right, so we set the table and let's have author's name. We said, let's indicate the country, indicate, cite the paper, all right, indicate the mean, and just get me the standard deviation. That was our target. So when I get the paper, we have the parameters we're looking for. I've listed them. I'll just get this paper, does it have this parameter? If it has it, I get the mean, I get the SD, put it on the table. If it doesn't have it, we exclude it. All right? As simple as that. So generally, you want to pick the author's name, publication here, study location, country, sample size, very important. You can't be doing a study and you omit sample size. In fact, one of the things the reviewer wants to see is this paper you consulted, do they have enough sample size for the study? Because some people do some studies and they don't consider sample size. So, and they finalize that area is everywhere with 6 sample size. And you want to use that kind of study for meta-analysis? No, it's not going to be fine. Right? So the last thing is that the data related to the outcome of interest, very critical. You can't be looking at maybe diabetes, for instance, and you are not taking data on insulin levels or blood glucose level. And you want to go take data on kidney parameters. It's a deviation from what you are looking at, all right? So let's look at some specific outcome of interest for different studies. For example, for prevalence studies, you may want, if you are doing something relative to prevalence studies, for example, you're looking at prevalence of COVID-19. You will not want to say in the whole world, because it's going to be very cumbersome. So you want to say South Africa or in KZN, you narrow it down, okay? And you look at, okay, what you want to look at in that paper is present. Is it absent in this study? What do they report? In how many patients COVID-19 they looked at sample size of 100? They reported present in 60 and not present in 40. So that's the data you are taking in a prevalence studies. And you look at the effect measured in the study. What are the effects measured? Did they measure heartbeat or heart rate? Was there blood clotting? Things associated with COVID-19, difficulty in breathing, sneezing, were there data on such parameters? Okay. For correlation studies has to do with mostly non-experimental research method. I'm used to understand linear relationship and association between two variables. So you have two variables and trying to compare their association. Okay, is there association between this particular compound and this particular disease, or this particular organism and this particular disease? So is smoking associated with Alzheimer's disease? It sounds funny because how does the smoking cause memory impairment or neuropathological effects? But as there are studies that has reported it, you can, okay, do that. So you want to get the author name, the sample size. The correlation studies, we always have an R value. Get the R value. The R value will be needed for your meta-analysis, okay? And which country, all right? Studies with continuous outcome data, these normally deal with numbers, numerical scale that you are able to measure most times. Maybe in the biomedical field, you're measuring blood glucose level. That's a continuous outcome. All right, so you get the mean. There will always be mean because you can't use one sample. The least you can use is 3. All right, and there are some studies that will say we don't want 3, we want 6. So which will give you very strong data. So you get the mean, you get the standard deviation. I'm used to this continuous outcome data because it's relative to my field. So what I'm targeting is the biomarkers, parameters I'm looking out for. I'm looking for the mean, I'm looking for the standard deviation in any paper that I've included in my study. If you're looking at effect sizes in control group design, which is actually related to binary outcomes and dichotomous outcome, this actually deals with studies with N out of N, maybe 30 deaths out of 100 samples. They reported 30 died out of 100 that had diarrhea. So you want to get the author name, if that condition, diarrhea, how many people had diarrhea, all right, how many did not have, okay, want to indicate if there is comparison with the control that you're using, all right. So that's a size control group design. It's also used for a case control study, right? Case control studies, you may want to get the mean, okay, you get the standard deviation or standard error, you get the country, if there's a control, you get the mean of the control, you get the standard error of the control. Now at the end of the day, you can compare with your statistical analysis between the control and the effects, those that add to the disease or what you are targeting. All right. But for cohort studies, odds ratio is very, very critical. All right. You get more information on odds ratio in the next session, but cohort studies deals mostly with study approach with participant over a period of time, measuring, okay, in this particular city, which between this year and this year, what kind of things affect them and things like that. All right. I want to run so we can save time as well. We also have incidence ratios. We want to look at the event that occurs, the timing of that event between the treatment and the control, or the diseased and the control. All right. So other data which you can include, depending on the study, like I said, sit down with your team, be specific. All right. Okay. So where to find data in the articles? After your materials and methods, the next thing is results. So go into the result. But when I get articles, I don't rush into the results. I want to look at what are they targeting? what did they indicate in the method they are looking out for? Because your eligibility criteria will show you the parameters they are looking out for. So it's very easy for you to get into the result, for you to pick them up. You are looking out for tables. Many will report in tables, some will report in figures. So you want to check the tables, you want to check the mean value, you want to check the R if it's correlational studies, you want to check the standard deviation, you want to check the number of present or absent, the prevalence studies, and N sample size, very critical. So you want to look at figures and maps as well. Okay. I'm rounding off now. So this is where you may want to use some software or application to be able to extract data. We're able to get this, one of the work we are doing currently. And we got to a point, some of the articles we identified did not indicate any table. So we had so many figures, histograms, line graphs. This is one of the studies. So I want to extract the data. Wanted to get what is the mean value there, because they didn't indicate it on the table. It was challenging, and we had already started the study. So when I was working on it, I called him and said, son is serious. I can't assume this. It will be difficult to assume, and I can't just say, let's pick this value and report it, because we'll be reporting something that is wrong, and we don't want to. If you are to query your work, or after 10 years somebody reads your work and wants to reproduce it, and is able to identify that the data you picked here is wrong, and writes the journal that you are spreading what is not correct to the research community. So we started searching online. How can we use application to extract data? So we got this web plot digitizer. There are many of them, and I'm happy to introduce you to this web plot digitizer. It's an open source application. You can also download it, right? That's where I'm going to round up this session. I opened it just now. Okay, this is it. Web plot. Let me share it for those that are online. So this is the web plot digitizer. I want to, sorry, the Zoom is covering. Okay, I think we can see it now. Let me bring this down. Okay, if you want to check it, just Google web plot digitizer. You can open it. So I went on YouTube, and because I was curious, we can't get stuck here. We started this work, we must finish it. I went on YouTube to find if somebody had worked on it, and I saw a guy who was able to display how to use it. All right, so it was a bit tedious, but getting used to it, and if you have passion for research, you will know that along the line, you'll get used to it. So you just need to click launch, and it brings you here. This is the interface. It shows you that you can use this to extract your data. What you need to do is just make sure you get the image of your data. So what I had to do is that I went back to that paper, the PDF copy. I screenshot that particular graph I want to extract a data from. All right. And I loaded it on this. Okay. So when you load it, let me load one of the screenshots that I screen grabbed. I'm rounding off now. Okay. Okay. Yes. So you choose whichever one you're dealing with. If it's a line graph, pick XY plot. If it's a 2D if it's a bar chart, you pick the bar plot. So you click align axis. Now this gives you a direction on how you are able to extract your data. You will see point one is telling you to name the point from where your scale of your axis is starting from. So you click here for point one and here for point 10. So I can click proceed. So you see this is the graph. So I can click point one. So it's able to pick the axis and it is able to measure it the same way with the same scale of your axis. All right. So this tells you that point one is 0. And if you look at it, point two is 140. So you indicate it so that it can pick it. All right. Okay. So after that, the next thing to do is to indicate your point. And for accuracy, before now, I could not accurately get this point. But for accuracy, you can see, just place your cursor here and it picks it. All right, for you to get the data for that point. Go to the next one and pick it. The next one. You must be careful that you get the focal point. If you don't get the focal point, it will pick the wrong number. Maybe below or above. So after that, you can say view data. It gives you the exact value on each bar. Are we following? So how do I get my SD value? Because I was like, we need to get the SD value as well. So I pick this and save it because that's the first point. And if you are very familiar with standard error, you will know that your SD is between one value and the other. That's the error bar, the one below and one on top. So I just need to repeat the same thing. I'll have to clear this, but you need to note down that value. So this is the lower data on that error bar. Okay. So for me to get the higher one, I just need to click this one. I don't want to go through the process again because of time. So let me assume you understand. So you start all over again, load the image, and click this point where the bar stops at the top. It gives you the higher data. Then you subtract from the lower one, then you get your SD value. Shall I do that? Okay. All right, let me do that. So it is assumed that you have saved this one. Okay. Let's say for bar 0 is 98.24. Let somebody know that for us. 98.24. Right? So I have to close this now. I'll have to reload that image. Okay. Just to start again. You can get familiar with the app and get a more faster way to do it. But just ensure you extract your data the right way. Okay. So I'll click the top. Yeah. Just make sure you get the focal point. All right. If you're much in a hurry, you will miss it. Okay. All right. And if you miss it, you have to start all over again. You have to reload the image again. That's how it works. Okay. So
Just pick one or two parts. Yes.
okay. Let me just click this to 1 and 2. It's not that tedious, but if you're not if you're in a hurry, you get it. So you can. Okay. All right. Sorry. Let me relaunch the hub. But it's better you get the idea and that's all right. So okay. Sorry, I didn't define the points before. Before I started, let me start again. Apologies, please. Okay. Line axis. 1, 2. Okay, 0 to 140. All right. So you can do your data now. So you say that we had 98.24 earlier, and we have 103.15. So just subtract that. That gives you your SD. So you can do that for the rest. The same thing applies to line graphs, all right? Let me go to my slide. The same thing applies to line graphs if you have something like if you have something like this, so and you can do it. It takes a lot of patience to be able to do that. So you can use any of the digitizers online, graph extracts, just practice around it, and you'll be able to extract your data. So please save your questions. There will be time for questions. I know you have a lot of questions, don't worry. We'll be able to answer your questions. So that ends the session for data extraction. I hope I've been able to explain it in a way you can understand. All right. Thank you.
Because of our time, I think we have to move to the next session. Doctor, you need to change as the host. HDMI. That's not HDMI. There's no. No,
Yes, I will. The name.
Ibilala. Sorry. I said, Kone HDMI. I know HDMI. Kone HDMI. Okay.
So, yeah. Which one is that?
Need to plug.
Oh, this is that not other one for what? Is it seriously low? No, it has more power. It has battery.
Are you sure?
Yeah, that's enough.
Okay.
Well, if there is need, we can.
That's
That's battery. So we can't step out. Just signal me if anything happens.
money. Okay. Hello. Okay. Please, we are starting the next session. We just want to demonstrate how to use SPSS 28, version 28. So do mental analysis. There are different software that you can use that most of them, the free ones require you to know how to code. Myself, I'm a fan of R or Python. So I don't like clicking. Write the code, run it, and I like to do it in a lazy way. Maybe you take a walk, then your result will be available somewhere else on your drive and on your system. So, well, for the purpose of, for the benefit of our people that are not used to coding, we want to use this, then you can use Stata. There is a module in Stata for meta-analysis. We also have a medical calculator, MadeCAD. We also have it, you can use it for meta-analysis. Sorry.
Online.
Which one? Yes. Yes. Okay, thank you. So you, depending on the software that you want to use, the data structure is usually the same, but you just know the right formats, the file format that your software will accept. Most software will accept CSV or Excel format or text file. So you just need to arrange your data. So what we thought before is we pick some of the articles we have assigned the other time that they are fitting for our study based on this working title. We go through it one by one or divide ourselves into groups. But there is no time on our side now. We wanted to do it, practicalize it, but we won't be doing it. Well, I have some data that we can practice with. Here, I have the data on, I think, on dichotomous binary data, the one of prevalence, and I think one of the continuous data. So all I need to do is, okay, let me go through the traditional way. Sorry, this is not my system. I wanted to get the link copy. Okay, I get what is happening. It's the extractor now. Let me copy it into a more easy directory. Okay, we'll start with, I think this one is a prevalence. Okay, we can check the data. Let's just look at it. I have it in CSV format. This is one of the data from published work. So now, if you look at this one, there are a lot of covariates. And it depends, the basic things and this is a prevalence-based study, but depend on what you want to answer. Why you need to have some covariates? In the case you have, sorry. Thank you. So here we have a lot of covariates to study. This first one is serial number, the author, the year, the positive sample, and expand it a bit, the sample size, the percentage. We don't need this for any analysis, just put it there. Then this is the extraction method. This is a viral work. is on a virus. So we need to look at it. One of the hypotheses in this work is that we want to know maybe the extraction method really affects the results. So when you are doing meta regression, all these are important. So then the DNA extraction, IG, and what have you. So we also have the country, we also have the continent. You can derive continent from the country. Then being a kind of wastewater related paper. So we also have natural water, like river, the fresh waters, like that. Then the concentration method is also vital in viral study, different concentration method for the virus, the absorption method, the absorption elution method, the ultrafiltration, the lights. So we have to look at it and use it as covariates when we are dealing with. I told us the other time that the fact that they look alike, that can be, they look homogeneous, but when you start your analysis, you can discover that they have a lot of heterogeneities. So you want to dive into it, which part of the paper or the experimental work contributed to the heterogeneity. It can be from the water source, it can be from the concentration method, it can be from the detection method. So we have a lot of detection methods. So you have to understand that. So let's just go straight into the analysis. Let's just demonstrate it in SPSS. Where is my SPSS? This one? Sorry, this one? No, I'm not. I want the SPSS. Sorry. Okay. Let me just. Okay, fine. So now we have our data in Excel file. You can enter it manually, which I don't advise anybody to do. Because you have it already in Excel. So why don't you import it? So when you want to import data in Excel, just go to File, Import Data, then choose the format. The one we are using is in CSV format. So you choose CSV. If you have it in Excel format, choose Excel, SAS, or Stata method. So now, because I've already copied the directory, I can open it directly, place the directory. I don't like. Now, you must understand the first line contains the variable names. So you need to specify it. Then if there are white space, you can ask it to remove it. This one is especially important when you are dealing with test file based data. So let's click on it. Now you stop sharing. Second, which one? Sorry. Okay. No, let's look at the data. Okay, fine. Now we have the data set already, but there is a problem. The SPSS could not recognize the type of the data. So for any data, you need to specify this. So
most numeric data should be specified as scale data. And if it's just name, you use ordinal or nominal, depending on. So for years in scale, so we specify, no, author is ordinal or whatever, or nominal. Sorry, I'm not a statistician. There can be error. Then this one is scale. This is also scale. What do you have for sample size? Sample size is scale it. Then we can leave these ones as nominal. We won't be using them since we are not conducting them. Okay, I can demonstrate it. We need to do meta regression. Okay, this one has string. Okay, let's leave it like that and see. Okay, next thing is go to analysis. And we can see the module, which is meta-analysis, as the first one. Now, meta-analysis, go. What is that? Okay, because we have no binary outcomes. Please, can you check for me your own interface? What are the options you have there? So now the challenge is this one cannot handle, we cannot handle prevalence study. That is what I just discovered. Can't handle prevalence study, but it can handle binary data. So we use that one as example. Are we okay? I'm so sorry. I'm not used to this, but I just discovered that it will be useful for us since we are not, we don't have access to. So we import another data. So we use the second data to demonstrate it. I'm so sorry. This one can work. So I want to open new Spss and close this. So we repeat the same process of loading the data. We are loading the PRP data. Let's load the second data. Yes. So we have, they are all in CSV. Let's just open it again.
Okay, we have the data also. Then we change the variables name just to, okay. So just to rename it, we leave, in this case now, the SPSS recognize some of them directly. Scale, there's an error here. So we use a name that resemble command in the, I wanted to change this. Okay, let me save it for us. If I'll be able to change it. So I've changed it. Let's go to the analysis. Then because this one is a binary data and it's raw data, then we'll choose this. Now we have this interface. We can see the success, that is the, what I call it, the positive. Okay, the study ID goes into, we can use this one as a label. Then we choose the positive, that is, I think from the data, the positive is, sorry, I didn't name it with the convention we are using. NETN means the sample size. So for the experimental, the event size is E. We put E into sources, and then group control. So then because it's a binary data, we need to specify it as odd ratio. You can put it as log, or you can just leave it what you want the set size to be. So you choose the log, odd ratio, or whatever the form you want it to be at the end of it. So you specify the model, is it random effects or phase effect model. Then you click on confidence interval must be included. Then keep them in the meta-analysis. If you continue, analysis. We also, sorry, I think the arrangement of my data is different from what is needed. Okay, let me look at it if I can see another option. Sorry. I like working with codes. So this thing should
code will have taken care of this. Area outcome.
Is it blocked?
Yes. Sorry, we have some challenge with the data. It looked like I didn't prepare the data. I didn't prepare the data for SPSS. So I don't think it's in the right format. So that is why we have the challenge for SPSS. So one of the things we need to know is that we need to know how the software needs to get it in. Just like I said, I'm used to code, and this paper was originally prepared for her. So I was thinking we'll be able to use it directly, but somehow it's not working. Well, what I can do is if we are not able to achieve what we want to achieve here, I will record the session, then we'll mail it to everybody. I'm so sorry that we find ourselves in this. Now, just looking at the data we have is not in the format. So what I want to do is, let me check this other one. We can use it. If not, I will do the recording, then we'll send it across to our participants. Then I will add to it the interpretation. I'm so sorry, because this is one of the reasons why some of us are here. And sincerely, I don't have SPSS on my system, so I have to use someone else's system. So pardon me for, I told you I don't like all this trouble. But if it is a code, putting it together and go and sleep.
Or can we just go to questions and answers.
Okay, Prof, why we can go to while I'm trying to.
Okay, thank you everybody for listening to this talk. I'm sure most of us we hear this not listening for the first time. What I want us to do now is questions and answers section. So if you have any question on what our two facilitators are talking about, you can ask the question. And before we're talking about how we're going to get a group going forward, are you still going to be giving them an assignment?
Yes.
You know what we want to do is that we want to have a group at the end of this training, this workshop. And we're going to send an assignment to you. And those that will be able to do the assignment will be part of our group. Because I think we can do a lot together. And we need people from different fields to work together. So you'll be sending the assignment to you. We'll have your email addresses and we'll hope to get a response from all of you. I don't know whether you have any question. Okay, what's your question, Abiola?
How do you decide on the number of years you want to cover?
Okay. Well, your consideration should be the first thing to do is to do your literature search first. If someone has not done anything on what you intend to do. So a good place to start is Google Scholar for PubMed. PubMed will tell you there are reviews. When you check the reviews, are they in line with your question? If they are not, fine. You can decide anywhere. But in most cases, it's good that your review is all encompassing without temporal restriction. So, when in a case, maybe in the last 5 years, someone has done a review and you want to carry out an update on it, you can decide to start where the other person stopped. So that is the two way.
Okay, I also have this question. You've been talking about coding is very, very important. Coding is very, very important. And I also want to be somewhere like, I don't want to be wasting my time. So it's good for us to master coding. And you talk about R and Python. So how did you master it? Because there was the time I went for one Python. I don't think I can still do it till now. So how did you master Python and R? You know, tell us, did you go for any training and everything?
Thank you, Prof. Just like I said when we started, everything we are doing today is self-taught. But the time I wanted to try to go for a formal training, it was 700 euros. So, and I didn't have the money, I didn't get bursary. Because part of it is that you can get bursary for it. But one thing I discovered that, please, when you are doing your PhD or your master, explore it as much as possible. They can still give you travel grant, they can waste so much for it. Well, after you have your DR, you are on your own. You are on your own. Now, they said to me, I have applied, they said you are overqualified. Whereas I don't know anything there. Because I have PhD, they said I'm overqualified. So now you can get a lot of opportunities. So it's the kind of, going back to the question, is self-taught. I don't like it to be, I'm not the fan of this. I read textbook. So just identify a good textbook, follow them. Some people are very good at using YouTube. You have a lot of tutorials. Videos. You have a lot of tutorials there. But the main thing is you must be determined. Then just like this, I read a book.
I think is a kind of quote from scripture. I remember, as a man thinketh, so is he. So if you think you can know it, you will know it. If you think you can't know it, then you can know it. So you need to, one needs to be dedicated, read a lot. By the time you might be doing something that you need how to do it, you don't know. Google search. When you Google search, you might see somebody that have a problem. Maybe some people answer the person. There are forums where you can get it. But for me, textbook is the best. So then you might think I'm more school.
Thank you. Any other? Okay, wait. Let's go to people online. Okay. Okay. Do you want to read it to us? Okay. Okay. The person online should note and just ask his or her question.
Okay, thank you very much, sir. Firstly, I want to appreciate the organizer of this program. It has been very helpful.
Can someone read the comments? Okay.
Okay.
The audio is fine. We can't hear the person speaking.
Maybe it's the person.
You can type the question and drop it.
Maybe the person.
Okay.
The person is speaking, but we can't hear.
Okay,
The audio is fine.
Okay, ma.
Regarding the question that she asked, I think it can also depend on the research question and the scope of the study.
Yes.
For example, if you are doing a systematic review on COVID, you can start from 2020 up until now. So you have to also consider some of those other parameters that is peculiar to your study. Then that can guide the
Thank you. Just as a, now when you are dealing with work that needs to affect policy, you need to cover everything. Most things that Lancet, all these they have interest on what can affect policies. I read sometimes is one person, I think it's Stellenbosch or UP, he did meta-analysis on the yellow fever, fractional vaccination, whatever. So because his result was so key, is affecting policy now. His meta-analysis, so they want it to be all encompassing. So it depends on the focus and the research. Thank you.
Okay.
I'd like to ask, number one question is there a limitation to number of papers to be involved in meta-analysis? Also, which number is too small?
Well, there is no limitation to the number as long as they meet the inclusion criteria. So then the minimum paper is 2. Yes, but when you submit it, some people will query it. But when you did everything, well, there's some research areas that are too specific or novel. You just want to open the eyes of people to it. Then you find out at the end of the day, you just have maybe like 4 papers. I've seen people that started with how many papers? I think like 1,005. They end up with 3. And that was what they reported. They published in the Latin. So it's not at least you can identify anything to you are fine.
Then my second question is like somebody like me now that I've been working with a lot of people. I already downloaded it. Like, how do I go on from this level now? How do I go on? Do I discard all that because I love this new method now? Or how can I salvage so many of the information that I know are there in all these people that I already had? Research topics.
Well, if you want to start with what you have already, you'll be doing more than necessary. Because it's better you start all over again, divine everything, follow it. It will be easier. Some of the papers you have might still be useful. But start afresh.
About subjecting our data to the kind of test or something, I don't know, to determine whether it is homogeneous or heterogeneous. What type of test are you really talking about?
Okay. When we are doing this analysis, if not that we have to do with data, interface there talking about how to test for heterogeneity in the data. There are a lot of criteria, the high score, the age, whatever. So you have it there included in the analysis. So I will master this. Then most of the problem we have is data structure. So if I get the data structure, I will record it and send it to us.
Like somebody like I have a paper that I have graphs that I would love to extract the actual numbers or figures from the graph. And how we are not together. So I would like to please include that in the video that you are going to be sending to us, so that we'll be able to do that too.
What Dr. Tozina covers is what you need.
Yes. You don't cover that. It's talking about standard error and standard deviation.
Okay, you can.
So what is data?
I think I can answer your question. Whatever you have learned now, like you said, go back. Irrespective of the data you gather, because most likely you don't have a focus for that, for research question. Formulate your research question. Focus the data you want to extract based on the research question. Let me give you an example. The paper he helped me with, we started with the volumetric analysis. And I knew that I was going to introduce a narrative and a systematic review. So we got it, we got a Prisma figure and all of that. By the time I sent it to 4 journals, the reviewers were reading the bibliometric part. They were not looking at the area I was talking about, the mechanism, the biochemistry involved. So I sat down back again. I wanted to go into meta-analysis. So I spoke to him. I said, let's go ahead. And he said, no. We'll have to start all over again to get into meta-analysis to do that. So I said, no, it won't be fine. So I sat down and I saw that I can write a systematic review that is comprehensive. So I went to the papers I used again, and I extracted the data on a table comprehensive showing the outcome, and I explained the outcome. And the next journal, accepted it. So just go back, extract your data following those rules, those guidelines we're talking about. It's going to be very easy.
Thank you. Okay, 2 more questions. Okay, that's a question for okay. But you've added earlier on. I appreciate this cohort will be a continuous thing, not a one-off event. But most of the decisions here, they are in isolation. It must be a continuous movement, not a one-off event.
We want continuous. We need fun.
Noted. Okay, one more question. Okay, there's one hand. Okay, online. Okay, can you hear us on someone online?
Hello, please, if you are online, can you type your question?
Allow me to say my appreciation. Not really a question.
Okay, Prof asked the host now.
Okay, let's okay.
Yes, you are the host. So we need to transfer this to
One of these TMI.
I don't know.
Okay, no one hearing me. Okay, Toby, okay, can you just unmute yourself? Can you hear Toby?
Yeah, I can hear you. Hello. Can you hear me?
You can plug it.
Hello.
So look at, please help me check.
Please, can you hear me? Hello.
Okay. You will be able to hear them, but we can't hear her because the mic is not. Okay, the mic is not connected.
Okay.
Hello.
Take the mic to Prof. System.
Okay. Who is the host now? Okay, so we'll be able to hear. The Prof has to unmute.
Hello. Hello. Hello.
Okay, should be fine now.
Hello.
Yeah.
Okay, thank you very much. I just want to give a testimonial. I want to appreciate the organizer. I want to appreciate Professor Tosin. I want to appreciate every speaker for this workshop. It has really impacted me in not a small extent. I've written a particular review article before in Journal of Construction, Building and Construction, which is Q1 journal, but I never knew anything about meta-analysis, systematic review. I always see state-of-art review and all that. But in a nutshell, I just want to say thank you for this insightful, informative workshop. And I know that I will be able to do something better than what I've done before. Thank you very much. God bless you.
Welcome. God bless you too. Okay, I think that's it. Thanks everyone for coming. We've come to the end of today's workshop, and I'm sure that we're going to be having a good. You are going to send the assignment to them, but you are going to send an assignment to know those that are really serious. You know, let me tell you something. You can see that meta-analysis is not a joke. You must be serious. You must be ready to work hard. But I can tell you, you publish good articles, and we have Dr. Kundayo in our group that will guide us. So if you are ready to work, we are really going to be working together, and Dr. Tosin is also going to be part of the team. So on behalf of the organizer, I'm the organizer. I want to thank all of you for coming today. I'm glad that you were able to make it. I also wanted to thank the sponsor, NRF, for providing funds for us to do this kind of thing. And I'm happy that we have an NRF that is always sponsoring us. When you sponsor academics, they do good things. They make impact in society. And I'm happy that I'm in South Africa, where they really sponsor researchers, and it's very, very good. So I want to give this small gift to Dr. Olasende. Come and have this. Thank you so much. It's a small gift. I appreciate you. Yeah. Dr. Ekundayo, thank you so much. Dr. Ekundayo joined my group last year, and I'm telling you that he has really worked very hard. I'm happy that he's with us at DUT, and recently DUT just renewed his contract, and I'm happy that he's going to be here with us. And we're